Hi, hi everybody. So we are really excited about sharing our project with you, and of course, the main part of this presentation is gonna be talking about our approach and how we are applying it to domestic animals. But at the end of the presentation, I also included some slides to bring some thoughts about how can we apply this to wildlife fertility control. This is not an area that we are actively pursuing, but I thought this would be a great opportunity for inspire that discussion. Um, and before I start, I also would like to mention that although I'm the one here speaking today, I'm speaking on behalf of our whole team at Epivara, including our CEO, Dr. Jacob, who is a professor at the University of Illinois, and he started this project in 2016. So with that, I would like to start by talking about our approach. And first of all, our ultimate goal is to develop alternatives to the current practice of surgical removal of gonads. This is, of course, a method that is widely used in companion animals, and currently the surgical methods, including gonad sparing surgery, are the only methods that provide or guarantees that the animal is going to be infertile during the whole life, so guarantees sterility. But there's a need for alternatives that are more accessible in terms of cost, but also in terms of the need for facilities, skilled professionals, and equipment. So we have more accessible techniques that can help tackle the homelessness crisis and overpopulation crisis that has only got worse after COVID-19. And castration or surgical castration is also widely used in food animals. This is different in every country, but in the case of the United States, all the male piglets and almost all the bulls that are raised for meat production are being castrated after birth. This is considered necessary for improving the quality of their meat as well as for controlling their behaviors. But it's mostly done with any sort of pain medication. So, of course, it raises concerns about animal welfare, and there's a need also for alternatives. And at the beginning of this project, we set a series of specific objectives for our treatment to accomplish. First of all, it will have to be non-invasive or have a low invasiveness, and of course, be safe for the animal. It will have to induce a permanent effect after just a single treatment, be affordable, control sexual behavior in addition to fertility, since this is one of the main reasons why domestic animals are sterilized. And we thought that we could achieve this by inhibiting the reproductive development rather than trying to inactivate a reproductive organ that is already mature. So how can we do this? Well, we know that after birth, the animals go through a period of reproductive development, of the development of their hypothalamus, pituitary gonadal axis. So this process starts way before puberty. And we know that kisspeptin neurons that are located in the hypothalamus control this whole APG axis. So kisspeptin neurons releases a hormone called kisspeptin, KIS1, that is the one triggering the release of GnRH, that in turn triggers FSH-LH, and that makes the ovaries and testes mature, and eventually, when the animal is adult, produce eggs and sperm. So our running hypothesis is that if we target this kisspeptin, and we do this during this critical time of reproductive development, we can make the reproductive system stay immature and therefore induce permanent infertility or sterility. Now, this hypothesis is based on previous research and our understanding of the sexual differentiation of the brain. This process starts also after birth and determines the difference that exists between the male and female brain. These are different at the anatomical, but also structural and functional level. And the process of differentiation is regulated by sex steroids. So actually the male brain is exposed to higher levels of estradiol after birth. This is because their testes can produce testosterone, and this testosterone in the brain is locally converted into estradiol. And this estradiol is really interesting because it can have multiple types of effects and even opposite effects depending on the specific area of the brain or the hypothalamus. So it can both induce neurogenesis and also cell death. And in the case of the anteroventral periventricular nucleus, the ABPV, so this is a nucleus in the hypothalamus where a key population of kisspeptin neurons is located. Estradiol induces cell death, leading to a lower number of neurons and lower size of this area in comparison to the females. Then we have this process by which males are naturally programmed to have a smaller ABPV and lower kisspeptin neurons due to that exposure to higher endogenous estrogens. So what we do is we try to replicate this process in the females by treating them with estradiol or estradiol prodrug, leading to a decrease in their kisspeptin neuron numbers. And so when the animals are adults, they don't have the trigger for egg release. And in the case of the males, we can emphasize this process by also treating them with estradiol or estradiol prodrugs, leading to a further decrease of that kisspeptin neuron population and also affecting their capacity of producing sperm. So we tested this principle initially in rats. In this study in particular, we used this prototype drug that we call EPB300. This is basically estrogen-based. So we treated the animals after birth, and we waited until they were adults to recover their hypothalamus and use in situ hybridization to be able to visualize the neurons that are expressing kisspeptin. So as you can see here in this, in the ones that were treated, they are almost completely depleted of kisspeptin neurons in comparison to the control animals. We confirmed these results also with PCR and with single-cell RNA sequencing. And then in the females, we saw that their ovaries were smaller than the control animals, and they didn't have CL, so corpus luteum that we can see here in the control, they are not present in the treated animals. This indicates that they are not ovulating. And these ovaries also produce less amount of estradiol. In the case of the males, we also have a significantly smaller testis size compared to the control animals, and their seminiferous tubules, like we can see here in the histology, are depleted of sperm cells, and they also produce less testosterone. And we did also some breeding trials to be able to confirm that these animals were not able to reproduce. More recently, we were also interested in knowing how the treatment is affecting the sexual behavior of the females. So in this study, what we did is that we treat the animals also after birth. And when they were adults, we surgically removed their ovaries and then induced estrus with administration of estradiol and progesterone. And then we did a behavior test by pairing them with a sexually active male. So in the control animals, even if they don't have ovaries because they have that external stimulation, hormonal stimulation, they still display this type of behavior, the lordosis, which indicates the sexual receptiveness every time that the male is trying to mount them. And instead, in the treated animals, they didn't show any lordosis behavior at all, even after the hormonal stimulation. So what we think now is that the kisspeptin neurons are also responsible of controlling sexual receptivity. So that is why with a lower number of expecting neurons, they stay, or they cannot display that sexual behavior, even independently of their hormones. So I would like to talk now on how we move from this concept into products for companion and food animals. So the process of veterinary drug development in a very, very simplified ways goes like this. After we have our proof of concept, we start doing pilot testing in the target species for confirming the efficacy and safety of the treatment. And once we have a strong evidence that the treatment is working as expected, is when we can move to do larger studies. These clinical trials are the ones that are required for regulatory approval. In our case, in the United States, that is regulated by the Food and Drug Administration, the FDA. And the animal testing goes in parallel with the drug development itself, from the first formulation until we manufacture the product. So for the proof of concept studies, we use a lab-made prototype. During pilot testing, we optimize a prototype under good laboratory practices. And once we move to clinical trials, FDA requires to use GMP product. That stands for good manufacturing practices. So basically, this is a product that is produced at a manufacturing facility, a larger scale, and under high standards of quality control and consistency. In our case, we tested different types of prototypes at the lab level. What they all have in common is that they are drug delivery systems that provide a slow or gradual release of the hormone. We know this is a key component for being able to cause an effect after just a single treatment instead of having to administer repeated doses over a period of time. And here I highlighted just two of the main prototypes that we tested, microspheres and pellet implants. But we decided to move forward our project with the pellet implants. And currently we have GLP prototypes, and we are in the process of scaling up and moving towards having a GMP product. So this is how this product looks for the companion animals. We have named this iSpay or iNeuter, depending on the applications on females or males. And is a small implant, 2 millimeters in diameter. They are loaded in this kind of injector that looks really similar to the microchip injector. And they are administered subcutaneously, but they are biodegradable. So after a few weeks, as they release the drug, they also disappear. They don't need to be removed. Our approach involving treating really young animals, as I was mentioning before, in the case of cats and dogs within 2 months after birth. I should say, though, that we are in the process right now of determining this window of effectiveness. So far in our pilot testing, we have only tested within 1 week after birth. So we did multiple pilot testing in female dogs initially, until we reached the current formulation. And in our last study in dogs, we treated 12 females and that we follow for 3 years. And during these 3 years, the animals stayed healthy, they were unable to reproduce, and they didn't show sexual behavior. Some of these dogs also, 5 of them were still following. We wanted to keep having incoming data that prove the continuous infertile status. And after these promising results, we decided also to test in female cats. So far, we have only done preliminary testing, but we have a good indication that also safe and is working for cats. So we are planning to start a larger pilot testing in cats in this upcoming July. And in the future, we also want to try to adapt this technology to males, since we have, from our rodent studies, we have the indication that it could work also in males. This is just a picture of two of our female dogs that were treated a few years ago. And this is how the product for the food animals and in specific for the male pigs looks. So we have named this product iNeuter S, and it's again a biodegradable implant. In this case, we loaded in this type of cartridge and this type of injector. So in the cartridge, we can hold multiple doses. Each of these contains a whole dose. And once you deliver one dose, the cartridge auto-rotates, so you're ready to deliver the next one. So basically, this is just a way of administering the drug that is more practical within the farm setting. This treatment is intended for male piglets within 1 week after birth. This is a really convenient time because it's the time when piglets are usually being processed in farms for other treatments and also for castration. And again, the treatment will degrade over time, but the effect will be maintained at least until the animals reach market weight. This is in the United States, the slaughter is usually done when the animals are around 6 months of age. I think it's important to highlight that the main reason why pigs are being castrated is for preventing board taint. So board taint is that disgusting taste that the meat, the pork, can have. Especially, it is more common in the meat coming from intact males. And this is because it's related to the presence of androgens. Actually, the main components that are responsible for board taint come from the bacteria in the intestine. One of them is scatol. But the androgens, such as testosterone or androstenone, they prevent or inhibit the metabolism of scatol in the liver, leading to a higher accumulation of scatol in the fat and higher presence of board taint. That is why, through our approach, if we can remove kisspeptin, we can prevent the maturation process of the testes, the production of androgens, and therefore prevent the appearance of board taint. This is what we tested in multiple pilot testing. Excuse me. So we have done over 20 pilot testing in pigs until reaching the current formulation. And as you can see here, their testes are significantly smaller compared to the intact animals, and they do not have board taint. They do not develop board taint. And we also, after these promising results in pigs, we also wanted to test in cattle. We did a first pilot test in both female and male beef calves. We followed them for 15 months because that is the age when they are usually slaughtered in the United States. But it wasn't successful. So we saw a delay in the puberty onset. In the case of the females, a delay in the estrous cyclicity, but the effect wasn't permanent. Now we are not discouraged, and we actually have recently started a second study in which we are making changes to the dose and other aspects of the treatment method in order to improve these results. So for the female dogs and male piglets, we have a good indication now that the treatment is working, and we have decided to start the formal process of obtaining regulatory approval. There's many different pivotal or clinical studies that we need to perform, and they fall under 5 main categories. So we have the CMC, that is, it's more about the manufacturing and control of the drug production itself. But then we have effectiveness, target animal safety study, and for the food animals, also human food safety and environmental assessment studies. Here is how our expected timeline for approval looks in the case of the female dog product. We have only had so far informal meetings with FDA, and we plan to start the formal process later this year and obtain regulatory approval by 2029. I have highlighted here some of the costs of these studies, just to show how expensive it is, and we're talking about millions of dollars. And for our pig product, we actually started the process over a year ago. We have had many meetings already with FDA, and we plan to start the first study also later this year. The timeline here looks a little bit longer, and that's because of the need of those human food safety and environmental safety studies, which are even more challenging, or they go even through a more rigorous evaluation by FDA than the efficacy and animal safety. So a big milestone for our pig development plan has been obtaining food use authorization from FDA. After showing preliminary data about human food safety, they are allowing us to sell the meat of the animals that we are treating under experimental conditions. So this, of course, reduces the cost of these studies while we are still collecting our samples and testing the product. So yeah, as you can see, this is a very challenging project, and especially as a small company, we need a lot of resources and partners, and I wanted to highlight some of those. First of all, at the core of this project is our R&D team, but we are informed through our leadership and our scientific advisory board, who bring expertise in biology and veterinary science, as well as in drug development and business management. Now, there are many other components of the drug development process, and that is why we need a larger partnership network. Our network includes the research centers where we actually do our studies, as well as the facilities where we manufacture our product, but also includes a lot of different type of consultants, including regulatory consultants and legal consultants for intellectual property. And we are also trying to build relationships within key influencers in the animal welfare and animal contraception space. And of course, funding. We have been lucky to receive a few grants from our federal agencies and also from our state. But most of our funding has come from private investors. We are currently also in the process of raising more funds as we are moving to this next phase of our development that involves clinical studies. And of course, these are more expensive than we have done so far. So for being able to attract these investors, we need to present the potential revenue of our application. So we have done some market analysis and customer discovery. This is just an example in the United States for female dogs. We consider the percentage of the dog population that could receive this treatment, and also the interest within different sectors in the pet industry of using this type of technology in order to determine a revenue and also a market penetration. And we have done similar assessment for pigs as well, considering the number of piglets that are produced and the different type of producers and their interest in using that technology. I would also like to highlight the protection of our intellectual property. We have been granted 2 patents in the US and 1 in the European Union. And we have applied for others, both in the US and other countries that are selected according to our market potential. Achieving these patents is complicated, and also maintaining them is expensive. And last, I would also like to highlight our publications. As a science-based company, we are committed to make our research public and have it reviewed by our peers. After obtaining our IP, we have been able to start publishing, and we will continue doing so as we finish more studies. So I have highlighted here some of our main publications for your reference in case you would like to know more about our approach. And this is what I had about our project. I apologize if I went really quick. I just wanted to give you a big overview of what we're doing and where we are in the process. But I would like to take also a few slides to talk about potential applications in other species, as well as for control of population. We do believe that there's a potential for applying this approach to many species due to the similarities in the way their HPG axis is regulated. Now, from our experience by testing in the species here that I highlighted in green, we do know that we need to adapt the dose, sometimes even the specific hormone that we're using, the release profile of the implant, and maybe most importantly, the age window, so the time when the treatment is going to be effective. Determining all these factors could be a matter of trial and error, but it helps to have some understanding of the animal reproduction to guide this decision making. Now, there's a big gap on this research in many species, especially when we're talking about what's going on during the neonatal period. And just to give you an example of how can we extrapolate the age window, we know that from our proof of concept studies that in female rats, the treatment worked when it was administered up to 21 days of age. That is the time when female rats are being weaned from their mothers, and also it is considered the end of the infantile period. These phases, neonatal, infantile, juvenile, are key developmental phases at many levels, including endocrinological. And there's difference in the concentration of hormones, as well as in the sensitivity of the hypothalamus and pituitary to the positive and negative feedback loops. So if we have this information from other species, we can extrapolate equivalent ages in those species. And beyond this challenge of translating the technology from one species to another, I also wanted to talk about more practical aspects of applying this approach to wildlife fertility control in terms of potentials and challenges. Of course, the main potential is the possibility of having a treatment that can cause a permanent effect after a single dose. These implants also have a low cost of production due to the low cost of the raw material. But as main challenges, we have the need of capturing the animals to deliver the drug, at least in this implant formulation. There are other drug delivery systems that provide this extended release of a drug, like the microspheres, that may be adapted for remote delivery because they are in a solution. But perhaps the main challenge is the need of targeting this neonatal juvenile population. And of course, in many species, we are not going to have access at all to those animals. If we would have access to them, targeting this population can actually maximize the impact in population control because we're ensuring that the animals never become fertile. And then there's consideration also about reproductive behavior, which could be interesting or not, depending on the sex and species. Similarly, we could consider adapting this approach for controlling population of free-roaming cats and dogs, and could potentially be integrated in the current programs of trap, neuter, return, with the added benefit of not needing to move the animal to a facility to do the surgery or not needing recovery. But again, the main challenge is to have access to that neonatal juvenile population. And in TNR programs, usually they try to prioritize adopting the animals that are trapped that young. Although, of course, that depends on the access to adopters or foster care. But the key takeaway is that there is potential, but there's also main challenges that need to be considered for each species. So with that, I would like to thank the Epivara team, as well as Dr. Ko's research group at the University of Illinois and our funding agencies. And thank you for your attention.